Well, hi everyone. So today we are in another soil seminar online this Friday. And today we have Edgardo Sepulveda, who is going to present about Rhizobacteria associated with Solanum hindsianum that promotes the plant growth and decrease the effect of Fusarium oxysporum in the tomato. But first, I'm going to give a short introduction about Edgardo. He's a biologist from the Faculty of Science and a doctor in biomedical sciences from the Center for Genomic Sciences, both at the National Autonomous University of Mexico. He held postdoctoral positions at the University of Tübingen, University of Illinois at Urbana-Champaign, and the Program of Protein Evolution of the Max Planck Institute for Development Biology in Tübingen. His research interests include the discovery and characterization of transcriptional regulators and the associated signal transduction pathways in bacteria, the isolation and characterization of plant growth promoting rhizobacteria, and the structure of the microbial communities associated with agricultural soils in Baja California. And presenting that, so now, Edgardo, you can start with your presentation, and thank you very, very much for being here today.
Thank you very much for the nice introduction. Thank you very much for the invitation. I'm very happy to be here. I really like to cross these disciplinary borders that sometimes we have. I think maybe this talk will be more useful for me than for you, but thank you again for inviting me. So as you hear, I'm more like a microbiologist. I am a molecular microbiologist. I am mainly interested in signal sensing in bacteria. But let me tell you the story about how we started working with plant growth promoting Rhizobacteria. So I live in Ensenada, a small city, 80 kilometers away from the United States border. And I work in CICESE, that is the Center for Scientific Research and Higher Education of Ensenada. This is one of the first, this is a public research center that is funded by the Mexican government and was one of the first centers that was moved or installed away from Mexico City. In Mexico, everything is very centralized. And at some point, like 30 years ago, they decided to start developing the rest of the country. And this one was one of the centers. So this CICESE was founded in 1973, and we do basic and applied research. We have three divisions: the division of Earth and Sea Sciences, which was the first one, the divisions of Physics, and the division of Life Sciences, which is the one in which I am adscribed, which is the youngest one. It's only 20 years old, not much. And we have 12 departments. I am part of the microbiology department. So in these public research science centers, scientists are not only supposed to make basic research, we're also supposed to make research that impacts the population around or in the regions in which they are settled. So we are always encouraged to do some applied research. And that's the reason why when I arrived, like in 2017 to Ensenada, I did not have only to develop my research about transcriptional regulation in bacteria, but I also have to develop some research lines in issues that are important for the population on the region. So this is a small map of Ensenada. We can see a very nice city. It's very large. It's like a long strip of land. Here we have the main population center. Here we have a small industrial region and a port. And over here we have the agricultural region of Maniadero. In the agricultural region of Maniadero, we have a lot of crops. A lot of crops are grown, and their main market is California. And the characteristics that these growers have is that they are organic certified, because in California, they like organic agriculture, they like organic products, they pay very good money, and there's a lot of money that come from growing different organic crops here and then send them 80 kilometers directly to the stores, and there's a lot of money to be made. No, we have every kind of crop you want to think about it, but the main products are Solanaceae, mainly peppers, tomatoes, some chilies. They also grow different herbs. everything you can think. We also have strawberries, different berries. A lot of things are grown here in Mañadero. There in the north, we have the famous Guadalupe Valley, which is our wine county. This is where almost all the wine production in Mexico is being done. Not anymore now, other regions of Mexico are also producing wine, but Guadalupe Valley was like the place where everything started. And then a little bit further in this direction, we have the Ojos Negros Valley, that is also another agricultural region where they grow also some grapes, but they also grow tomatoes, they also grow strawberries, they grow watermelons also, and they also have some cows and do some cheese. Not a very good cheese, but everything else is really good, really delicious, really high quality. We often don't get to eat anything of the things that are grown in Ojos Negros or in Mañadero, because Mexican economy cannot afford the prices of organic agriculture. So we just see the trucks going up, and sometimes when something doesn't pass the quality checks, then we are able to buy it. And this is only the closest region to Ensenada. The reality is, maybe you think about the Baja California Peninsula and you think about this desert, the big desert full of cacti. And yes, we have two very big deserts, but also we have a lot of agricultural land. This will be the municipalities. Ensenada used to be the biggest municipality in Mexico. Now it has been cut, and now it's has been cut in half. So now it will be the municipality of Ensenada, and the other half will be the municipality of San Quintin, which is also a huge agricultural region where you can find principally berries and tomatoes. Also, everything organic, and everything goes directly to California. Up here, we will have Tijuana. Tijuana is not very agricultural, but Mexicali is very agricultural. They do not grow tomatoes, they do not grow fruits. They are mainly growing grains. So it's a completely different kind of agriculture. No? So we people at CICESE that are in the municipality of Ensenada have established good relationship with the producers of our municipality. So we have good interactions with grape producers, we have good relationship with tomato producers. In general, the surface that can be or that is cultivated in our state is 70,000 square kilometers, and there are 78 different crops that are being grown, tomato being the main producer. So we have a strong relationship with tomato producers. Tomato is a plant that is very sensitive to plagues and diseases. It has high nutritional requirements, so the producers each cycle have to apply up to 35 agrochemicals like fungicides, insecticides, and fertilizers. Of course, this doesn't happen if you are a certified organic producer. You are not allowed to use this kind of agrochemicals. No? Among the principal diseases that our producers face when growing tomato in Baja California is root rot, the mildews, late blight, Fusarium wilt, some cankers, nematodes are a big problems, and viruses were like a silent problem that they have, but since CICESE got recently hired a virologist, now we are understanding how big is the viruses in Baja California agricultural problem. So the thing with us is that we were approached by a producer that has a lot of problems with Fusarium wilt. And what is Fusarium wilt? Fusarium wilt is a disease that is caused by a fungi that is called Fusarium oxysporum, from a species Lycopersicum. There are different races because, you know, several plants have been developed to resist to this disease, and of course the microorganism has adapted, and you can find different races that will cause the same disease. No, the symptoms of this disease are very clear: the yellowing of the leaves, and most importantly, a narrowing in the crown, in the region where the roots and the stem encounter each other. There's necrosis there because of the invasion of the fungi, and after a while you have also smaller size of the plants, and also an important reduction in the production. And sometimes the plants don't even get to produce flowers, they will die earlier. So this is a big problem. This producer had a problem with this disease. We went to his land. We took some samples. We isolated the causal agents for the disease. He had indeed Fusarium oxysporum forma specialis lycopersicii, and we isolated 2 different races causing this disease in his land. We found race 1 and race 2. We haven't found race 3 in Mexico yet, but race 1 and race 2 are common problems in Baja California. So everything that I will show you was done specifically with the causal agents that we isolated from this producer. So as I told you, these producers are certified organic, so they are not allowed to use strong chemicals. They still use some chemicals that with organic, there's a manual, and if your chemical is in the manual, then you're allowed to use it. But there are very little chemicals, and of course, what they are doing is using plant growth promoting Rhizobacteria to compensate and to as an alternative to these agrochemicals. No, they use different kind of PGPR. Some use endophytes, some use rhizospheric bacteria, and these bacteria have the ability to induce plant growth and inhibit pathogen activity by different mechanisms. Some that are direct are, for example, nitrogen fixation, solubilization of nutrients, or the production of pheromones that will induce plant growth, that they will induce root development. And some of them are indirect, like, for example, the biocontrol of pathogens, the production of siderophores that will also antagonize pathogens, and the induction of plant systemic response. Just the presence of a bacteria that will not produce a disease anyway will activate plant systemic response, and when the pathogens come, well, the plant is already ready to face. The problem that we face often, and these producers in Baja California face, is that often when they inoculate with the commercial produce they are sold here, they will not produce the desired effects, neither in growth promotion or in disease control. They just apply their stuff that they are sold, and it will never work. There's a huge economy on this kind of products here in Baja California because we have organic agriculture, and often they will bring some product from Israel, some from Europe, and they apply them, they see no response. Why? Because the colonization and the activity of plant growth promoting bacteria depends on several factors. For example, the weather, the physical properties of soils, the existing microbiome, and plant bacteria communication. So you may apply a lot of bacteria, but maybe they will not be able to survive even for 1 hour. Maybe they will be able to start to get established in the rhizosphere of the plant, but they will not exert their plant growth promoting properties. There's a lot of different reasons, no? But mainly it's because the bacteria will not be able to establish within the plant. They will not be able to colonize. And in this fact, plant bacteria communication is important. And often it has been shown that the plants are, the role of the plants shaping its microbiome is really, really important. With all this work now with microbiomes, it has been shown that the plants will form their own microbiomes. And if you compare plants of the same family, the microbiomes will be more similar among more phylogenetically close plants that those that are less close. No, so for example, we have this analysis with the Posea, in which we can see here how the plants that are the microbiomes of plants that are this part will be made of mice. This is a Teosinte, which is a wild relative of mice. And then we have wheat and sorghum, and we can see how their microbiomes are closer of the plants that are more phylogenetically close. So this will show that similar plants will select for similar microbiomes. So what we thought is, well, people are bringing these bacteria that were isolated from, I don't know, carrots in the Netherlands, and they are applying them to tomatoes in Baja California. So maybe this is the reason they are not working. They just can't adapt to the soil. They cannot adapt to the plant. So they are not working because of this. So what we thought is, let's use a close relative of the plant that we are interested in to treat and get plant growth promoting rhizobacteria from them. So we choose Solanum hindsianum. Is a plant, a Solanaceae that grows in Baja California. It's endemic to Baja California and the Sonoran Desert. This plant grows under arid conditions. It has resistance to drought, and of course, it's a Solanaceae. So our idea was we want to apply or to get plant growth promoting rhizobacteria to be used in the tomato production. So then let's go and get a native Solanaceae that is adapted to the conditions of the region, and we will hope that because of this, the bacteria that we get from this plant will be able to adapt and colonize the tomato, and will be able to promote plant growth and to inhibit the Fusarium wilt in them. So then we made a very nice trip, several trips throughout the peninsula to collect them. You can find the Solanaceae, the Solanum hindsianum or Mariola. We will call it Mariola from now on, everywhere. If you go a little bit south of Ensenada, you will start finding this plant in the correct season. You will find it just growing near the road. So you just go with the car of the institute and until we found them. And we collect the plants, we collect the roots, we collect the whole plant from all these different regions. This will be the state of Baja California. This will be the state of Baja California. And in all of these places, we collected some of these plants. Then we go back to the lab with the plants, and we will take the rhizospheric soil and make several dilutions to isolate some bacteria, but we will also take the roots. We will surface sterilize them with some alcohol and fire, and then we will crush them to get the endophytic bacteria. So we tested several media, several growth temperatures, and then we plate them, and then we will just pick bacteria with different phenotypes. And in total, we were able to isolate 343 isolates. At this moment, we will just focus on the rhizospheric bacteria. And from the rhizosphere, we were able to obtain 343 isolates. Then we took these 343 isolates and screened them for two different characteristics: plant growth promotion and antagonism against Fusarium oxysporum. For the plant growth promotion, we use colorimetric tests to test for different activities that are associated to plant growth promotion, like siderophores production, chitinase production, cyanhydric acid production, indolacetic acid production, and phosphate and potassium solubilization. In this way, we could just use like 6 plates, because we tested each strain by triplicate, and we will just stamp them, and in the matter of a couple of weeks, we were able to identify which characteristic each of our isolate had. Then after we did this screening, we selected those with a lot of we selected those that presented at least 3 or 4 characteristics, and we made a second round of screening. This time we tested for biofilm formation, nitrogen fixation, zinc solubilization, sorry, this should be a C, and ACC deaminase production, that are also characteristics that are associated with plant growth promotion or with pathogen inhibition. And we also now quantified the production of indolacetic acid. Before it was just a quantitative assay. Now we quantify it, so we could select those with the better characteristics with the characteristics that will suggest that they will promote plant growth or will inhibit the pathogens. So from the 343 screenings, we selected these 6 bacteria. We sequenced their 16S RNA gene. So we were able to identify them. Almost all of them belong to the Pseudomonas, but we also found a Paenibacillus and a Bacillus strain. And we decided to use these 6 strains and test them for plant growth promotion. As a control, we use non-inoculated plants, but we also use the commercial product that the producer that contacted us with this problem was using, that is called Bacillus. It was marketed as a plant growth promoting and pathogen inhibition product. When we read the literature from the product, we discovered it was not a plant growth promotion product. Really, it was just a pathogen inhibiting product. But this happens often. The people that sell these products will just go to the producers, will tell them whatever they want to hear, and they will just buy it because they are so desperate that they try everything. They really, really, really try everything they are being sold. So to make these experiments, we did 8 repetitions for each treatment. We germinate the seeds after surface sterilization. In pit moss, they were germinated for 30 days until they got their first true leaves. After this, the plants were transplanted to 6-inch pots. Everything was done under sterile substrate. And 7 days after the transplant, the day that the plants were transplanted, they were submerged in a solution with a concentration of 10 to the 8 of the plant growth promoting bacteria that we selected. And then they were transplanted, and then 7 days after the first transplant, they were re-inoculated, this time with 5 milliliters, also of a solution of 10 to the 8 of the plant growth promoting bacteria. Then the plants were set up in our greenhouse, and they were left there for 55 days after the last inoculation. They were watered often. This work was performed by a master student that is called Carmen. She's a very nice agronomist. So she will take care of them using all her training, looking or growing them like they will be grown in the greenhouse by the producer. So after the 50 days, they were evaluated, plants were evaluated for different plant growth promotion indicators like plant length, stem diameter, dry weight, number of flowers and leaves, for example. So these are our results. There were no difference in flowers or leaves, sadly, but we have very clear effect of our inocula in stem length, stem dry weight, stem diameter, and root dry weight. So we can see here the control, and we can see that it's significantly smaller than the plants were significantly smaller than the plants that were inoculated with our isolates. And as expected, the product that the producer was using was not as good as our isolates. But this is not the fault of the company that made it, because they clearly say this is an antifungal agent. It's the fault of the vendor that told the producer that he will also get plant growth. No? But anyway, we can see very clear effect. So the bacteria, that rhizobacterial bacteria that we obtained from the Mariola, were able to promote plant growth in tomato. Then we focused now in the antagonism against Fusarium. Again, we did everything with the 343 isolates. This picture is a small example of what we got. This is very clear. This is the actinobacteria that we found. Yes. And for this, we make first a big screening against Fusarium, oxysporum, Formes specialis lycopersicus race 1. We did this because race 1 will grow faster than race 2. So at the end, it's a matter of, we have a lot of isolates, we have to test them. So we need a fast way to test them. So we selected the race 1, and we will put 4 isolates in each side of the plates, and we will see if there was some inhibition. Sometimes there was no inhibition, like here, where the fungi grow over the bacteria, but sometimes we will have very clear inhibition, sometimes by the formation of an inhibition halo, and sometimes because the bacteria will just invade the whole plate and will inhibit at the end the growth of the fungi. After the quantitative assay, we did the qualitative assays. We did quantitative assays now, this time with the race 1 and the race 2. This is the race 1. We can see that the races look very, very different, but they will cause very similar disease. Sometimes we will get both races from the diseased plants. Sometimes we will get only race 1. Sometimes we will get race 2. But these two fungi were there and in this land, and they were breaking havoc. So here we will see how the fungi will behave without the bacteria, and then here we have some examples of how the bacteria were inhibiting this fungi. Then we cross the information from the inhibition with the information from the plant growth promoting activities to select those bacteria that will be possibly more useful. And we selected these 8 isolates. A lot of them were Bacillus. We also found some Streptomyces. But I should say that a lot of the Streptomyces isolates that we selected were not that nice to the plants. They will kill the plants sometimes. So we focused on the bacilli instead of the Streptomyces. So at the end, we selected 3 Bacillus strains and 2 Actinobacteria, the Arthrobacter and the Streptomyces luteverticulatis, and we perform also in vivo assays. So as a negative control, we use just non-inoculated plants. Then also as a negative control, or just as a control, we use again the commercial product that the producer was using. As a positive control, we were using plants inoculated only with fall air1, race 1 and race 2. And then we did also the plants inoculated with the pathogen and with our agents, inoculated with the pathogen and with the commercial product. Again, we did 8 repetitions of heat treatment. Everything was made just like with the plant growth promotion assay, with the difference that 7 days after the second inoculation, we performed the infection with fall race 1 and race 2 at the same time. So we will grow the race 1 and the race 2 in rice independently, and then we'll just inoculate a small scoop of the infected rice inside the soil where the plants were growing, and then we'll just with a steel spatula we'll make some holes in the soil, and in this way we will make some wounds in the plant so that the fungi will be able to more efficiently invade the plants. Again, 60 days after evaluation, we measure different indicators like plant length, stem diameter, dry weight, numbers of flowers and leaves. Again, we found no difference in numbers of flowers and leaves. As a fact, I think we should have waited longer so we could see differences in number of flowers or even in fruit production. But anyway, here the results were not as dramatic as with the plant growth promotion. In blue, you will see the plants that were not inoculated with fall. In red, you will see the plants that were inoculated with fall. So if you see the first column, you will see the control, the plant that was not treated with any biological control agent. And you can see that the inoculation with the fungi will have very strong impact in all of the measurements we were doing. It will affect stem length, it will affect stem dry weight, it will greatly affect stem diameter, just as we expected for this disease, and it will also affect a root weight. The only indicator in which we see no difference was in root length. And then in some of the plants that were inoculated with our isolates, we could see that this difference between the infected and non-infected plant will disappear. Normally, when you see this kind of graph, you will look for the indicators of statistical significance of the difference of the means. In this case, we don't want to see this. We want the inoculated infected plant to behave just as the non-inoculated plant. And we could see that several of our biological control agents will have a very good effect in which you will not distinguish the infected plant from the not inoculated plant. No, especially this isolate, the isolate RBES262, it has a consistent inhibition of the pathogen and the effects of the pathogen in in tomatoes. So this was a good candidate for the control of Fusarium oxysporum. We gave this agent to the producer, and he has been using it for a while. The original deal is that we were going to be able to go there and monitor, but sometimes when you work with producers, you have to understand that you have a scientific interest, but they also have a commercial interest, and a lot of people depend on their activity. And sometimes after they got what they want, the communication is not that, doesn't work that good anymore. No? So we know they keep using it. Sadly, we have not been able to monitor in field anymore the activity of our inoculum. But well, at least we know it worked, and it keeps working, and they keep using this. This was a Bacillus isolate. So then we have these very good results. We have been working with other producers. We work also with vine producers. We keep using native plants now. We are using these native vines, or not native, but heritage vines now, to get some plant growth promoting rhizobacteria for the wine industry. It also has been working very good, especially when we work against phytopathogenic fungi. But at the end, our hypothesis was that these bacteria were going to work because they were adapted to the environmental conditions, and because we were getting them for closely related to the crops that we were treating, no? And we were still wanting to test if our hypothesis was right. So we wanted to know if the bacteria that we obtained from Solanum hindsianum were really capable of establishing in the rhizosphere or to colonize the plants, the crops. Because with plant growth promoting rhizobacteria, you have to understand something. We are always sell this golden standard in which you will inoculate a bacteria and it will stay there like for 6 agricultural cycles, and your producer will not need to inoculate anything because now it's there. But the truth, that's not the case. It's never the case. The producers will always try to reinforce it. We will always reinforce it. We found better results when we're inoculating continuously. So we don't know if the bacteria are working because they can colonize, or if they just work in a very specific moment of plant development, and then it doesn't matter if they get lost. But we wanted to test our hypothesis that the bacteria will be able to colonize from Solanum hindsianum, will be able to colonize the crops, the Solanaceae that were agriculturally important. So we decided to test this using 3 different crops that are grown in Baja California: tomato, serrano chili, and tomatillo. And we selected 2 Pseudomonas from our collection. This one, the 087 from the rhizosphere that I already showed you in our plant growth promotion results, and the EPCA E10 Pseudomonas strain that I haven't shown you in any result, but that we obtained, that is an endophyte from the Mariola, and that was also tested from some plant growth promoting bacteria characteristics. We selected these Pseudomonas because Pseudomonas is much more easy to work with. It's not that easy to transform and to do genetics with Bacilli, with Actinobacteria. So the Pseudomonas strains will be easier to work with. So we took a rhizospheric Pseudomona and an endophytic Pseudomona from Mariola, and we intend to test if they were able to colonize these 3 different crops. To be able to monitor them, we tag them with a TN7 transposon. It's a transposon that will insert in a very specific place in the Pseudomonas genome. So we will know that the insertion of the transposon will not cause any havoc in any gene. It will not affect the bacteria in any way. It will insert at the end of a transfer RNA outside the open reading frame, so nothing will be mutated. And this TN7 had a GFP protein. As a fact, we have a big collection of this. We have these isolates tagged with different version of GFP with different colors, but we selected the classic GFP because it was the one that gave us better fluorescence. We can see that the fluorescence was not exactly the same in both isolates. So this will the 152. This will be the endophyte, and this will be the rhizospheric. And then what we did was to inoculate plants, different 8 plants of each crop. The first experiment, we did it in semi-axenic conditions. We made a substrate from 75% soil from a farm and 25 pit moss, but then everything was sterilized. We surface sterilized the seeds also. We sterilized the substrate. We sterilized the water in which they were grown. And they were inoculated with a much more lower amount of bacteria. We will only inoculate directly in the substrate. We will apply 1 milliliter of a 10-to-8 solution of the suspension of the bacteria. So we will inoculate the plants and we will keep them for 60 days. And after that, we will process the rhizospheric soil, and we will also collect epiphytes and endophytes. We will make serial dilutions, and then we will grow them in kanamycin media, because the transposon also have a kanamycin resistant gen. And then we will leave them under blue light, and we will be able to see clearly which were our which bacteria belong to our bacteria, which were our bacteria. And even if we when we did these semi-axenic conditions where we try to keep everything very clean, we obtain a lot more bacteria. Not only our bacteria grow there, because we suspect a lot of the bacteria came already inside the seeds of the plants. And well, these are our results. And we can see that our bacteria were very successful colonizing the 3 crops. They all were able to colonize tomato rhizosphere, chili and tomatillo rhizosphere without any problems. And the most interesting thing we found was that only the bacteria, that the endophytic bacteria was able to get inside the plants. The bacteria, the isolate that we collected from the rhizosphere never got to get inside the plants. It was never found as an endophyte. Only the endophytic one was found as an endophyte. So we see no difference. We also use 2 different regimes of watering. We use one in which we will water 50 milliliters a day, and the lower will be water 20 milliliters a day. Again, because since they were, these bacteria were isolated from a plant that is used to growing under a drought, we thought they will be able also to withstand low watering conditions. No, and we basically saw no difference between the watering conditions under this, the conditions that we tested this. But then we say, okay, we did everything semi-axenic. The bacteria did not have to face any challenge. Let's repeat this as it will be made in the field. So this time we did not sterilize the substrate. So the 75% of soil will be colonized already with the bacteria that were growing in the farm. The pit moss will be sterilized. It's sold sterile, but the 75% of soil will be colonized. We will not sterilize it. We did surface sterilize the seeds still, but we use non-sterile substrate, as I told you. And also these plants were just watered with tap water. We never sterilize the water. So every time we water, the bacteria will be washed. The bacteria will be renovated. We will get whatever was in the water in the moment we water the plants. Again, we use 2 different watering conditions, 100 milliliters 2 times a week, or 50 milliliters 2 times a week. And this time we just put them in our greenhouse in an open stage instead of a growing chamber. So everything will just fly on top. Other experiments will probably contaminate them. So the bacteria where our isolates will not have such an easy time this time. No, they will face some competition. And yes, these are our results. We were not able to get the results for tomatillo in time. They are already finished, but they have not been processed yet. And we see that the bacteria this time have a more problem colonizing the plants. They face competition, but still they were all there at the end, maybe in lower concentrations. Biggest difference is that in high watering, the rhizospheric isolate was not able to grow in the plants as an epiphyte, neither as an endophyte, but that was expected. But under the low watering conditions, the rhizospheric bacteria was able to grow as an epiphyte again. So this will show that at least our hypothesis that this bacteria will be better adapted to drought conditions was true, and that the bacteria was able to regain its place in growing as an epiphyte. No, and again we saw this very strong difference in role. The bacteria that we originally isolated from the inside of Mariola was able to colonize the insides of tomato and chile, but the bacteria that we isolate from the rhizospheric soil was not able to colonize. So we know that all endophytic bacteria were rhizospheric bacteria sometimes, but we also know that not all rhizospheric bacteria are endophytic bacteria. No? So these results prove our theory to be true, that the bacteria that were isolated from the wild Solanaceae were able to colonize the agricultural relevant Solanaceae. No? So this work was done by these three excellent scientists. Carmen was the scientist that started everything. She's what we call a fires agronomist. She's a very good agronomist. I'm sure when she finishes her PhD, she will be poached by some important growing group. She isolated all the rhizobacteria. She characterized everything that I showed you before the transposons was made by her. Ana Corral, she did all the experiments with endophytic that I did not show you because I will not have had time, but we got very similar results to those of Carmen. And Jenny that made the experiments for testing the colonizations of the isolates. Everything in the context of a global pandemic. So I'm very proud and I'm very grateful for their job. I just want to show you now the rest of my group. Here we have Carmen. She did this project as a master student. Now she's a PhD student. She still works with plant growth promoting Rhizobacteria, but for vines. And now she's really good. She's mentoring undergrads, for example. No, this will be Carmen. This will be Sam, her undergrad student. This is Jenny that will defend her master thesis next week. Ana Corral now is working in a company that produces produce, working specifically with tomatoes. So she moved to the professional life. And Celi, Gemma, and Indira are working more in my transcriptional regulation topics. And well, that's all what I got for you today. I'm happy to answer any questions you will have.
Thank you very much, Edgardo. That was a great presentation, a lot of work, so that's amazing. So now we are open to questions. I have Stacy who has a question now.
Yeah, thanks for that presentation. It was really interesting. And it was nice of you to introduce your institute and everything, because I don't know much about that part of Mexico and all the agriculture. So that was really nice. I was super interested about the comparisons between Chile and tomatillo and tomato, and whether or not sort of those patterns that look like tomatillo, the slide you showed, it looked like it was more easily colonized by the bacteria. And I wonder how it compares just in terms of disease and such. Like, is tomatillo worse than tomato in terms of disease, or is it tougher?
Tomato is the weakest plant of all of them, especially against this agent. We did extensive testing with Fusarium to try to standardize our pathogenicity tests, and it was really difficult to get tomatillo infected. Tomatillo was really, really resistant. Chili was not easy, not difficult. I will not say it's difficult, but tomato, you can just look bad at it and it will get sick. So it's a very clear, sometimes as a fact, when our producers are having problems, they will just rotate crops. And they will rotate when they start having a lot of problems with tomatillo, they will move to Chile. And then after Chile starts getting problems, they will move to tomatillo. And that's how they keep working. And they consider, in general, tomatillo to be a stronger plant against this disease. Against a mildew, for example, is not that resistant. It will get diseased very, very easy. So my experience is with this disease in particular. With other diseases, it will not replicate.
And do you think it's just that the tomato varieties are really inbred and tomatillo is not so inbred? Or do you think it's something else like morphological or biochemical that's inherently different between Physalis and Solanum?
Well, I think that the problem is more like evolution, because for a long time, the thing that was grown in Baja California was only tomato. Tomato, 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 tomato. So these pathogens have been selected for 20, 30 years to infect tomato. And it's more a problem of the monoculture than a problem. And so I think it's a combination of the genetic homogenization of the tomato and also strong pressure, selective pressure to infect tomato for so many years. Now, things are getting more organic, people are trying to rotate crops, and we hope that this will help, or to make a strongest enemy, but at least give them some space to work.
Oh, thank you. That was really helpful.
Welcome.
Thanks, Stacy and Edgardo. Any other question? Okay. We do have a question from Deidre Jacobson. She asks, do you know if the inoculation has an impact on leaf defense or insect damage? Actually, I was going to ask something about that, because
this will happen sometime in the University of Baja California, just 250 meters away from my lab. We have a college which we are working with, is Dr. Rafael Bello Bedoy. He works with herbivory by insects in Solanaceae in native chilies. And we have plans to start working on that to see if the inoculation of the plants with our bacteria will have an impact on the grazing by insects. But we have not tested it yet. It's in plan. We have a student, but then she needs to leave, and the project is right now in pause. But it will happen someday.
Yeah, related to that, I was wondering also if you have seen any systemic changes in the leaves or the aerial part of the plants in this species, because it's just a long time ago I was checking that, like the effects in antioxidant system, for example, and so other species, but it was something different, was soybean. But
yes,
the population of Rhizobacteria was actually inducing the systemic changes in antioxidant reactive oxygen species in leaf and stem.
Yes, and that's exactly what Rafael does. So when we manage to get a master's student or a PhD student to work on them, we will be able to answer it. We want to answer that. We need to do it, but still we haven't been able to do it. But it's in our mind.
That's awesome. And then we have Federico Roda with a question.
Hi, Edgardo. Nice to see you again.
Yeah, hello, hola.
Edgardo, I have one question. So you mentioned that for these inoculations to succeed, it was important to get microorganisms that came from the same environment and a closely related plant. So do you think that according to your results, the phylogenetically related is more important than the same environment? Or because it seems like some of these things were isolated from Solanum, but they work kind of better in tomatillo, which is physalis, rather than tomato. So maybe at this scale, it's more important just the environment, or what is your take at that?
Well, we have these results for colonization, but we still have no results for plant growth promotion. So we don't know if they are better in plant growth promotion in chili and in tomatillo. We are doing experiments. A lot of them are already finished, but Jenny needs to graduate, so we haven't tested them. Until now, it seemed that the plant growth promoting activity is better in tomato, but we cannot, we have to be very careful with these results because the plants are very different, and it's very different to compare them after 60 days. Okay, because for tomato, for the variety we're using, 60 days is more than enough to almost start growing fruit, but chili will already be full of flowers, and tomatillo will have nothing. The plants will just be getting ready to produce flowers. So we are really not completely sure. What we know is that the plants are able to colonize. That's the only answer we could get until now. At some point, we would like to be able to cross this information about plant growth promoting with rhizospheric, with colonization, but we are still not there. We still need to process the data. And something important is to remember is that we as biologists are thinking like ecologists. We want to think about colonization, about the effect, but at the end, the producers, they just want them to produce an effect, and it's difficult to perform this kind of experiments with them because they will just apply them. They will just apply them. They don't care when this happens. They just want the product to have an effect. So we don't know yet when these bacteria are important. Do they really need to colonize the plant the whole time for them to have an effect? Or maybe if they are just needed at the beginning, we still don't know that. We would like to get to know that at some point. I don't know if we will be able to know this, but still, I don't think we have enough information to answer your question.
Thanks a lot, very interesting.
Gracias.
I hope to see you again.
Thank you, Fede and Edgardo. So, it's just a data. I think that great talk, really interesting and beautiful diagrams of your methods too. Yeah, that's great.
It was made by my students. Everything is made by my students. My work is just to discuss with them, to plan with them. But these very nice graphs and everything is the work of my students.
That's awesome. Okay, so we don't have any more questions, so we are finishing this seminar today. Thank you very much, Edgardo. And in 2 weeks, we'll have the last seminar of the year. That is Vanessa Morales from Chile, who is going to be talking about Schizanthus, the backstage of synopsis and future perspectives. So thank you, everyone, and see you.